Biology Part 173. Well, our tutorial describes the germination of the broad bean seeds and consider formation of specialization of tissues in stem. Well, our tutorial describes the adaptation of roots and leaves, seed structure and germination. The broad bean belongs to the plant group known as the Dictolidans. Its embryo can be observed by peeling off the outer coat or the testa of a seed which has been soaked in water for 24 hours. It is a simple structure consisting of a single axis bearing two large cotyledons and seed leaves. It shows the embryo within the cotyledons pulled apart. The sections of the axis above the below junctions with the cytoledons are known respectively as the epicotyl or as the hypocotyl. The tip of the apocotyl, called the pilmate, grows to become shoot, and while the tip of the hypolose radicle gives rise to the roots. Most seeds, include those of the broad beans, become dormant when first formed. That is, they pass through a period when germination cannot take place. Even if external conditions are suitable, the mechanism of dormancy are varied and complex, and it will describe in the tutorial. Dormancy is broken to the exposure of the cold, peritoneal drying, or a variety of other environmental changes. And sometimes you need to have the favorable temperature, oxygen. In the first stage of germination, a broad bean seed absorbs and inhibits the large amounts of the water, swelling to more than twice the original size. Next, we will have the meristems and differentiation. The meristems, unlike animals which can produce new cells from growth, the replacement is most part is their bodies. Plants have separate specialized regions called the meristems, where the new cells of the plant body are formed. The most important meristems are the apical meristems, which occur near the extreme tips of the shoot and root, and the tips of the sides of the branches show the structures observed at the tip of the shoot of the dictylodon. This apical meristem also gives rise to a small leaves which grows to enclose and protect the apex of the shoot, forming the apical bud, axillaries, axillary buds, a node, exogenous, and internodes. We, in fact, sometimes reach to the position where the regions of maximum growth is expressed and demonstrated. The differentiation is mainly concerned with the growth of the cell walls. These structures, although non-living, are the most characteristic and visible feature of the plant cells. They are secreted by the living part of the cell called the protoplast, while the cell is still increasing in size and consists of many cellulose. Pectin is there, the glycoprotein is there, cellulose synthesis occur at the glycoprotein, and so on. Between the walls of the adjoining cell is a thin layer of sticky pactic material called the middle lamella, which binds the cells together. This structure is first formed during the division of the meristematic cells when the vessels of the Golgi body comes to a midline. Although the developing cells are bounded by their cell walls and separated by the middle lamella, two features prevent them from being physiologically isolated. Firstly, the cell wall is porous, allowing the free exchange of dissolved material. Secondly, there are thin regions where the wall is penetrated by the threads of cytoplasm. And of course, there is plasma desmata is there, and there are pits, and there are primary cell wall laid down during the development from merisematic cells.